KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video we are discussing prokaryotes information point of view. Remaining information we have to discuss ribosomes and one more is inclusion bodies. So related to the prokaryotic cell, so remaining information, what is information to be discussed now? Ribosomes and inclusion bodies. Once this particular information is complete, so we will be entered into the eukaryotic cell information. So now let us start to discuss ribosomes and inclusion bodies which are coming under, means actually ribosomes are present in eukaryotes also, but here I have given information regarding prokaryotic ribosomes information. So they try to understand clearly, try to differentiate carefully. Right. Now ribosomes. So in prokaryotes, ribosomes are associated with the plasma membrane of the cell. So that's we know very well know why, because so this particular point in previous video also I have given. Okay. So in prokaryotes, ribosomes are associated with the plasma membrane. Suppose if you take a prokaryotic cell, what is the layer just below the presence of or below of the cell wall, we will be taking as cell membrane, isn't it? So that particular cell membrane consists of, or what are the ribosomes are there in prokaryotes, definitely are associated with the plasma membrane. Okay, so now there are about, so what is the length or what is the size of these ribosomes, means there 15 nanometers to 20 nanometers size they have. So definitely what is the size of the ribosomes, means 15 nanometers to 20 nanometers in size. And up to here these two lines we have learned in a previous video, but no problem, now in this video also let us revise. Next one, and are made up of, means here these ribosomes are made up of with two subunits, made up of two subunits. So one is 50S and 30S. Sir, S means what, sir? Means Swedberg unit. Swedberg unit or sedimentation coefficient. So by the process of centrifugation, so sedimentation coefficient means based on the size or based on the molecular weight, so we can separate the ribosomes. That's why here this one S is the units of ribosomes. Simple. If physics is there, means in a CGS or MT, likewise SI units or CGS units, likewise units are there, so likewise here ribosomes point of view, unit is S. Okay, now so in prokaryotes, so two subunits together, means two subunits, one is 50S and 30S, or so two subunits are there, which when present together form 70S type of ribosomes. Sorry, once you see there 50S ribosomes and 30S ribosomes together, 80S dana, sorry, here you have written wrong, sir, don't say like that. Okay, so here 70S type of ribosomes subunits or 50S and 30S. Don't add mathematically. So if you add means 80S will borrow. So 50S and 30S, so actually 80S comes, but 80S illa, that is correct one is 70S. So means 70S type of ribosomes, now we have been divided into two subunits, one is 50S and one more is 30S. My dear students, here nulla concentrate panikunga. Okay ma. So in prokaryotes 70S type of ribosomes are there, 70S type of ribosomes, two subunits are there, one is 50S and one more is 30S. Next one. So here the 70th type of ribosomes are prokaryotic ribosomes. But in previous video, so in I think so in the second video for cell the unit of life, no. So there one point we learned 70th type of ribosomes. 70th type of ribosomes not only present in the prokaryotic or not only present in the prokaryotes and which are also present in chloroplast and mitochondria of eukaryotic cell or eukaryotic plant cell. So suppose if you learn this point there, no worry, or otherwise now you learn here. What is the point? 70th type of ribosomes perfectly, so we can see in prokaryotes, and same 70th type of ribosomes we can observe in eukaryotes also, not in cytoplasm, where we can observe in chloroplast and one more is in mitochondria. So thereby, my dear students, now here if you take eukaryotic cell, listen carefully, now one more time I will revise that statement. Listen now. If eukaryotic cell is there means there in a cytoplasm, no, we can find out the presence of 80th type of ribosomes. Now only we learn now 70th type of ribosomes where it is present in eukaryotic cell means and are all present in where? In a mitochondria and in chloroplast. So thereby in a eukaryotic cell, two types of ribosomes are present. 80th type of ribosomes are present in cytoplasm and 70th type of ribosomes are present in where? Chloroplast and mitochondria. And so here that is a point we have to revise. Next one, these ribosomes are the site of protein synthesis. So here these ribosomes, what are the ribosomes are there? No, definitely these ribosomes are helps in the process of protein synthesis. So thereby idu nalla remember pannikunga. So because of the presence of ribosomes, so what is the process we can observe? Protein synthesis. So definitely here the protein synthesis process is going to be taken place means definitely chance is there of the presence of ribosomes. Next one, several ribosomes may attach. One more time you see that. This is important point. In prokaryotes, no, this is important point. Now, several ribosomes may attach to the which one? A single mRNA. So there are several ribosomes are there. So there are several ribosomes are there and attached to a single mRNA. Ipo romba means there are different number of or so many number of ribosomes. Several. That's why here the several ribosomes are attached to the single mRNA and form a chain. So that particular chain we are calling as a polyribosomes or polysomes. Polyribosomes or polysomes. Now here, sorry, on a single or on a single stranded of which one mRNA, we are observing the presence of several ribosomes. Suppose here one mRNA is there means here we can observe the presence of more number of. Here we can observe the more number of ribosomes presence on the mRNA. So there when more number of ribosomes are present on the mRNA, no, definitely that particular structure we will be calling as polyribosomes or polysomes. So now only we seen, sir, why sir so many number of ribosomes present on the mRNA sir means I think so it is high standard, whether you know or not I don't know. So what is the use here? We have seen proteins, ribosomes use protein synthesis. So how protein synthesis process takes place means, so here the mRNA consists of a message. M stands for message, messenger RNA. So what is the message that particular mRNA consists of? So in the presence of ribosomes, that particular message is translated to proteins or translated as proteins. So simple, ribosomes are there. These ribosomes of a polysome translate, once you see, translate the mRNA into proteins. So here translate the mRNA into proteins. Okay, now you see. So here polyribosomes are there. So these polyribosomes we are calling by one more name, polysome. Okay, now here once you say, now here what is the use of these ribosomes means we have seen here protein synthesis. So thereby, so ribosomes of a polysome translate the mRNA into the proteins. I told you, no, mRNA means messenger RNA. So here the message will be there. So based on that particular message, no, so proteins formation takes place. Okay, so that the entire proteins formation synthesis of proteins, the entire process will be under the control of ribosomes. Once you see here polyribosomes, a small diagram here I have drawn. Yellow line is there, no, yellow color line, adi mRNA. On the mRNA, no, so now we are observing, once you see there, மேலே கொஞ்சம் பெருசுதோ கீழே கொஞ்சம் சின்ன முட்டை மாதிரி இருக்கணும். ஸோ அது large subunit, small subunit together present on the mRNA. So this kind of structure we will be considering as polyribosomes. இப்போ polyribosomes, so poly means many dana. So many number of ribosomes are commonly present on the mRNA. So that particular structure we will be taking as polyribosomes or polysomes. Next one, this is somewhat information regarding ribosomes. Next information is inclusion bodies. Inclusion bodies point of view, reserve material in prokaryotic cells are stored. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. So thereby, my dear students, what is the point? So what is the reserve material? Reserve material which is present in the prokaryotic cell, no, definitely stored where? Stored in the cytoplasm only in the form of inclusion bodies. So what may be that reserve material? Reserve material is extra material. So suppose here, suppose if the bacteria takes any kind of material, no, so suppose if that particular material being like a extra, so extra, so means excess. Okay, so that particular material will be stored where? In cytoplasm only in the form of inclusion bodies. So next one, these inclusion bodies are not bound by any membrane system. So means here, why? Because suppose whatever may be that excess material, so is not bound by any membrane system. So membrane system likewise nothing. So nothing will be there. So or surrounding of these inclusion bodies. Next one, live freely or live free in the cytoplasm, or lie means present, lie freely in the cytoplasm. Okay, for example, what are the examples we will be taking means, so phosphate granules, cyanophycean granules and glycogen granules. So there are different types of granules are there. Phosphate granules, cyanophycean granules, glycogen granules. Likewise, there are different number of or different types of which one inclusion bodies are present. Next one, vacuoles. What is the kind of vacuoles? Gas vacuoles. Gas vacuoles are found in the blue, green, purple and green photosynthetic bacteria. So this particular point also, examples, neat examination point of view important. Okay, so gas vacuoles are found in where blue, green, purple and one more is green photosynthetic bacteria. So then what is the use of these gas vacuoles? Means filled with gas definitely helps in floating. So that particular point also here we can save in our mind. So means after completion of inclusion bodies and ribosomes, no, complete information of prokaryotic cell is completed. So ribosomes information and inclusion bodies so completely comes under the prokaryotic cell. Now let us start the information of eukaryotic cell. Eukaryotic cell point of view, the eukaryotes includes, we have learned classification point of view, there are 5 kingdoms are there according to 5th kingdom classification proposed by R.H. Whittaker. So what are those kingdom? Monera, Protista, Fungi, Plantae, Animalia. First one means kingdom Monera definitely comes under prokaryote. Remaining all means second kingdom to up to 5th kingdom means which one? Kingdom Protista, Fungi, Plantae, Animalia, all kind of organisms except kingdom Monera. All organisms we can be taken under eukaryotic cells. So இப்போ பாருங்க, so eukaryotes include the all protists, plants, animals and fungi. இப்போ you see there all kind of kingdoms covered, no? So here you see protista cover ஆயிடுச்சு, fungi, plant, animalia. So all these plants or all these organisms we will be taking under eukaryotes. Next one, in eukaryotic cells, in eukaryotic cells there is an extensive compartmentalization. So here definitely, so in eukaryotic cells, there is an extensive compartmentalization of the cytoplasm. Means here this particular cytoplasm is divided into so many compartments. Ipo suppose in our home is there, means so kitchen ki apram praying hall, apram sleeping hall, dining hall, and likewise different rooms are there in one house. Likewise in one cell, so different compartments, means for cytoplasm that can be divided or many parts. Suppose because of the presence of different types of cell organelles, chloroplastic, mitochondrial, endoplasmic reticulum, Golgi complex. Likewise, so many number of cell organelles are there, no? So because of the presence of so many number of cell organelles in the eukaryotic cells cytoplasm of the eukaryotic cell is divided into extensive compartmentalization through the presence of membrane bound cell organelles. Point purida. One more time, once you see, in eukaryotic cells there is an extensive compartmentalization of cytoplasm through the presence of the membrane bound cell organelles. Easy information. Because of the presence of cell membranes or because of the presence of cell organelles, cytoplasm divided into many parts. Adidama simple da. Next one, eukaryotic cells possess an organized nucleus, well organized nucleus. Well organized nucleus means, what is the point we have to study? So well organized nucleus is nothing but the presence of nuclear envelope or nuclear membrane. Next one, in addition, eukaryotic cells have a variety of complex locomotory. So definitely, suppose if you take, means along with this entire information, so somewhat extra information also is there. What is that? In addition, eukaryotic cells have a variety of complex locomotory. So here the different types of complex, means in a locomotion point of view, definitely different types of organs as well as organ systems are present. Not only complex locomotory and the cytoskeleton structures. So here the cytoskeleton structures, so definitely you are present. These cytoskeleton structures which are also we can be seen. So which can be seen only in case of prokaryote, only we can be seen in eukaryotic cells. So 1 minute you see, listen. Eukaryotic cells, cytoplasm. In eukaryotic cells, cytoplasm is divided into extensive compartmentalization. So because of the presence of cell organelles. Next one, well defined nucleus is present, nothing but presence of nuclear membrane. Next one, in eukaryotic cell, complex locomotor structures we can observe. Along with all these things, we can observe cytoskeleton structures. Cytoskeleton, cyto cell, skeleton, so skeleton. So what is the use of this cytoskeleton? Means cytoskeleton meaning of first pathokoma. Cyto means a cell. So what is a skeleton like structure which is present in the cell? We will be taking as a cytoskeleton. Even in our body also, bones are there, skeleton structure. Means in our biology labs we can observe skeleton. That particular skeleton, even in our body also, if you see there, that particular skeleton is completely surrounded by the tissue. So thereby one particular structure, like one particular structure of the organism we are observing. Means here likewise, suppose if a skeleton is not there, means bones arrangement is not there in our body, means so our parts cannot be differentiated. Okay, so likewise here also skeleton like structures which we can be seen in a cell, that particular structure we will be taking as cytoskeleton. I hope you understood this. Next one, their genetic material is organized into chromosomes. Within the nucleus we are finding the presence of genetic material. That particular material, genetic material, is organized into chromosomes. Means here definitely in eukaryotes no we will observe the presence of chromosomes. So why? Because now only we are learning genetic material is organized into chromosomes. Next information, now continuation information. So all the eukaryotic cells do not are not identical. One more time, all eukaryotic cells are not identical. So their plant and animal cells are different. Okay, so now only we are observing no. So all particular cells will be not like a same. So definitely differences are there. Suppose if you observe the plants and animals differences no there. What are the main important differences we can find out? No, listen, differentiation is important. Suppose if you take a plant cell, no, plant cell consists of for possessing of cell wall, plastids and a large central vacuole. Listen carefully, important. Okay, so in a plant cell point of view, cell wall is there, plastids are there. Plastids means if chloroplast is there, no, chloroplast also comes under plastid. While explanation of plastids information, I will give you entire information, no worry. What is that? Plant cell wall or plant cell consists of for possessing of cell wall, plastids and large central vacuole, so which are absent in animal cells. If these three structures, cell wall is absent in animal cell, plastids are present in animal cell and large central vacuole also is absent in animal cells. Suppose if you have plastids means we no need to cook rice and everything. Okay, so definitely if you stand in the sunlight, no, definitely food materials preparation process takes place. But here we are not having plastids, no, that's why we are struggling for food. Okay, so all these needs or all these works for what? For obtaining of food only, no. So likewise here animal cells are not possessing of plastids. And on the other hand, if you see there, no, animal cells have centrioles, so which are absent in almost all plant cells. So animal cells, no. So what are the main important structures we can observe means centrioles. So these centrioles which we will not be finding in almost all kinds of plant cells. Almost all kinds of plant cells. So this is somewhat a differentiation. Suppose if you would like to learn the information regarding the differences between the plant cell and animal cells means or pakkathale characters podugonga keele one by one cell wall, plastids and vacuole and centrioles. So first three are present in plant cells and the last one is present in animal cell. And the last one is absent in plant cell. Okay, likewise on a table madri you can differentiate. Okay, next one, let us start to discuss the one by one cell organelles. So what are the compartments or what are the cell constituents are present, no? So one by one let us have a look. Now first of all let me explain regarding cell membrane. Cell membrane or another name is plasma membrane. Now definitely, so unknown point, so that is one or not unknown, very well known point. What is that? So this particular cell membrane is present just below the cell wall. So just below the cell wall. So in previous video we see no, there definitely this particular cell membrane makes the or that is associated with the nature and that will be connected with the outer environmental conditions. Now let us see the information regarding cell membrane. Now the detailed structure of the cell membrane was studied only after the advent of the electron microscope in 1950. Even year also remember pannikonga, important. Means when we have started to study about the structure of a cell membrane means we have started to study about the structure of cell membrane only after the discovery. Advent means discovery. After discovery of electronic microscope or electron microscope in 1950. So meanwhile chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possibility or possible structure of cell membrane. So means here So here what is the structure? No, the scientists they thought to, or they would like to find out what is the chemical composition or what is the chemical substances which are present in the cell membrane. They would like to find out. At the time, no, what they have taken, what is the material they have used for their experiments means, so they have been taken RBC cells, human RBC cells. So thereby here when they have used these RBC cells, no, they have studied or they have explained. Deduce means explanation. So they have given explanation. So possibility structure of a plasma membrane. So means here they may ask you, so what is the ideal material for studying of cell membrane? Means ideal material for study of cell membrane is RBC cells or human red blood cells. This is important point. Save pannikonga. Next one, these studies showed that the cell membrane is mainly composed of lipids and proteins. Again important point. So when they have started to study, they are learning one by one continuously. Now what they have studied, now they are giving one point to us, that is this particular cell membrane is mainly composed of made up of. So it is composed of lipids and proteins. Okay, next one, lipids point of view, a major lipids are phospholipids. So there in a lipids point of view, phospholipids are present there. That are arranged in bilipid layer or simply bilayer, BIM, bilayer. Bi means how many? 2. So that definitely, that's why here what are the lipids are there, no, or the phospholipids, these lipids are arranged in. Once you come down, so I hope you are observing this particular diagram. So here once you see there, this particular diagram is appearing or not? Once you see there, so here one here motta madri chinna spherical structures we see now. So these particular spherical structures we will be calling as a head, and the head ulle 2 lines or we have drawn, no, ingama exactly at spotlight, these structures are tail. Okay. So now here the head is there and ulle tail is there, and keele also once you see here the motor like structure spherical shape zero madri irukku, is structures head da. So here head is situated outside and tail is there towards inside. Okay, ఇది కొంచెం సేవ్ పనుకోంగా, నెక్స్ట్ ఎక్స్ప్లెనేషన్ డిపెండ్స్ ఆన్ దట్ ఓన్లీ. Now here now major lipids are phospholipids arranged in 2 rows. So arranged in 2 rows, instead of that one we are writing like bilayer. Bi means rendu only, no, 2 layers. Next one, also lipids are arranged within the membrane with the polar head. This is diagrammatical explanation, listen. Now here also lipids are arranged within the membrane with the polar head towards the outer side. இப்போ பாருங்க, இது polar head which is situated towards the outside or outer sides, and hydrophobic tail இப்போ ரெண்டு lines are there, no, இது hydrophobic tail towards the or situated towards inner parts. Definitely you have to remember this one. Okay, so polar head or simply polar or non-polar adu pakkathali pottom, but heads towards the outside and definitely tail towards the inside or inner parts. Next one, this ensures, so sir, why sir, suppose if it is being like that or if the arrangement being like that, then what is the use, sir, means. So here now we are learning, that is this ensures that non-polar tail. So what are the structures are present within the bilayer? No, these non-polar tails of saturated hydrocarbons are protected. Okay, non-polar tail of saturated hydrocarbons is protected from the aqueous environment. Okay, so it means here the what are the tail regions are there? No, these tail regions are present within the bilipid layer and protected by the aqueous environment. Next one, in addition to the phospholipids, so here we have seen, no, lipids are there. In lipids also majorly phospholipids are there. Likewise we see, no, in addition to the lipids means phospholipids, so this particular cell membrane also contains cholesterol. One time மேலே வாங்க, இங்கே பாருங்க, mainly composed, means here the cell membrane mainly composed of, firstly we are learning lipids and proteins, they have given information. If lipids point of view, they are giving deep information. What is that? In lipids, no type of lipids they are giving, phospholipids are present, they are giving. If along with the phospholipids, again what they are giving, no information. Along with the phospholipids, cholesterol also is there. If totally how many types of chemical compounds we seen? So first of all lipids we seen, aprom proteins we seen. If lipids and proteins, along with the lipids and proteins, we are finding the chemical structure. One more is cholesterol. Emma, next one, later biochemical investigation clearly revealed. So in now they have started biochemical investigation. So in a biochemical investigation, what is the information they are giving us? So that one clearly revealed that the cell membrane also possesses proteins and carbohydrates. If proteins mele we seen, here proteins we seen. If along with the proteins, lipids, cholesterol and one more compound also is added, that is carbohydrates. Likewise, in a chemical or biochemical investigation, what is the point we are learning? Not only all those components, along with proteins, lipids and cholesterol, we are finding the presence of carbohydrates also, or carbohydrates also are involved in the making of cell membrane. So likewise, one important information we are discussing in cell membrane. So still lot of information is there related to the cell membrane. So that entire information to be discussed. So in the next video definitely I will continue the cell membrane. So thereby my dear students, request what is that, no? So while watching this video, don't skip anywhere. You cannot understand if you skip once. So now this is a structure, so rough diagram of cell membrane. So here in a future classes, no, definitely I will come with fluid mosaic model. At the time, detailed information of cell membrane, what are the functions or what are the activities, so everything, so in a detailed manner I will explain. So take care and be safe at home.